So that's me, Roger Meacock. Natural Healing Solutions has been my name of my practice since 2000. So moving on from PCR, we're going on to the modRNA jabs, which are inherently dangerous. So they are not vaccines. They are gene therapies, and that's included in their BioNTech SEC filing. They recognised it there. So it's not me saying it, it's actually in the company that developed these vaccines' own literature that they recognise that they are gene therapies. And I call them modRNA because it's not mRNA as we get in our cells. They've actually changed one of the bases in them so that it's not natural. So I call them modRNA just so that everybody knows what I'm talking about. As they redefine, when you've changed, they changed the definition of vaccine in order to be able to include these. And I say to people, if I redefine all small, furry, four-legged animals as dogs, it won't make my cat bark. Okay? So the lipid nanoparticles, which is what they used to deliver the modRNA into the body, they can cause inflammation themselves. They can cause emboli, so that's blood clots through clumping and allergic reactions. And there's various different mechanisms they can do that. They do not stay at the injection site. Anybody who's given their cow antibiotics in the backside to treat mastitis knows where you put it in is not where it stays. The idea that any oil-based injection would stay at the site of injection was complete fallacy. And we know that from all the other injections that we give, that they go body-wide. So especially they go to other organs which have got high fat levels, which are the gonads, so the ovaries and the testes, lymphatic tissue and the brain and the nerves. It's been shown now that the modRNA can enter the nucleus and can reverse into the genome, and actually the lipid nanoparticles make that 100 times more likely that those RNA, DNA will go into the cells, because that's their whole function, is to deliver what's in them into the cells and through membranes. And so the fact that we've got the cell has got a membrane and the nucleus has got its membrane. So the lipid nanoparticles will actually deliver far more, 100 times more into the cells than if it had been just injected without it, which is why they're there. But they've also shown this contaminant DNA that's in there also gets equally absorbed at that higher rate as well. So due to the pseudouridine, which is one of the bases that they've changed, the body doesn't always recognize it, and it doesn't stop the protein manufacture. They've now found that those who've had COVID jabs, 709 days in some people, they're still finding spike protein. So probably that isn't the original spike protein. It's probably because it's reversed into the genome, into the nucleus, and they're actually still producing spike protein on an ongoing basis. So they'll also, you're putting in strands of this modRNA, and of course the body will break it up, and that's what they've always said, that it will be broken up. But of course those shorter chain lengths actually have some biological activity themselves. They're called microRNA. Humans have about 5,000 microRNA, which are cell signaling, so they tell the cells and different instructions to the body what to do within the body. So the fact that you're putting something in which can be broken down into these microRNAs, and they've got no idea what they all do. They know probably about what 5 of them do. So if you've got 5,000 there and you don't know all their interactions and how they actually work together, they won't be working in isolation. You would need some sort of computer the size of a room to actually try and calculate how these will all work together. We've also got a prionogenic risk. And that's been shown to happen for the modRNA itself, and also potentially the proteins that they are instructing the body to produce. And there's been no work done to check whether any of the proteins which are instructed to be produced have any prionogenic activity. There's also a potential autoimmune risk. So if the proteins which get decided to be produced actually have any similarity to proteins which naturally occur in the body, antibodies to what they think is the pathogen will also then start to recognize self as a problem, and you'll get autoimmune problems happening. So the modRNA in animals, because this is my concern, is obviously where they're going to, obviously I'm concerned about what happens in people, but I'm also concerned what's going to happen with our animals. So there is currently one modRNA product for pigs in the USA called Security. And it's actually been ongoing since 2018. How they got it past the vaccination definition before the definition being changed, I don't know, but it just shows the level of corruption in the authorization processes. So Security is a platform, as a product platform, not a specific end product. So if I tell you what happens, so you've got an outbreak of, argument's sake, respiratory disease in pigs. The vet comes along, they swab the pigs, they send the swab back to the lab sequence it, they choose a gene of interest, and they make a modRNA vaccine for that, and then they send it back to the farmer to inject. Guess how long it takes from swab to coming back to the farm as a vaccine? 8 to 12 weeks. But you can't check anything, especially things like prionogenic activity, in 8 to 12 weeks.
Can you say what prionogenic means, please?
Yeah, I'll come on to prions in a minute. So prions are basically what cause BSE. So Creutzfeldt-Jakob disease, they are misfolded proteins which are infectious themselves, but what they're like seeding a crystal layer, once they go in to and start this misfolding process, it causes all the proteins around them to do it. So it builds up a plaque, if you like, and that's what then causes the neurodegeneration that causes a spongiform encephalopathy. So you get the brain and nervous tissue looks a bit like a Swiss cheese with all the holes in it, and that's why it's called that. And it's caused by these prions. So the declared intention is to move animal vaccination to modRNA jabs. It hasn't happened in the UK yet. That's one positive. They have an inherent risk and they aren't being checked, like the prionogenic activity. If a chosen protein or the selected modRNA itself is prionogenic, then it will initiate the protein folding and the prion formation. Prion shedding can affect the microbiome and then the environment. If an infected animal is slaughtered before symptoms manifest, the prions will be in the food chain and could potentially make meat for food effective for humans and animal carnivore products themselves. So dogs and cats, obviously carnivores, so they should have meat. And so the RNA and prion hazards. So the modRNA itself, as I said, can be prionogenic. The chosen protein can be prionogenic. So you get frame, and this happens because of frame shifting errors in terms of how the protein is made from reading the RNA that's gone in. And you can also get different areas on the protein have the potential to be prionogenic that they don't look at. So looking at it from one aspect of a single immune response, but they forget to check or look at the other parts of the protein to see whether they could have a different action elsewhere in the body too. So these proteins may be shed in the milk, sweat, urine, feces and saliva to the environment, and the prions may also shed as well. Prions in slurry used as fertilizers can go to the soil and soil fauna, so like worms and beetles and all the rest of it potentially. And the latest research shows that plant roots can uptake prions from the soil and accumulate it in leaves, which obviously if you're eating a crop, you're going to be eating it in your plants as well. So those of you who are perhaps vegetarian or vegan think that you've got nothing to worry about, got news for you, unfortunately, potentially you could have. So the prions in the soil will leach down into the water system. They're not neutralized by normal water processing. They can potentially spread, maybe same as microplastics can in rain. Who knows? I don't think the work has been done to show that. There's been a lot of question marks over how these, especially chronic wasting disease in the States is a big problem at the moment. But it started around some of the government laboratories. I'm not quite sure how that happened or why. But so this is just a diagram of what can potentially happen. So you've got the modRNA, and it obviously gets the body to produce proteins under instruction. And then you've got this PRPC, which is the normal form of protein in the body, and the action of either of them can make it go to the scrapie form. Scrapie is the sheep version of mad cow disease, the spongiform encephalopathy. And of course that could then potentially go to the topsoil, could potentially go to the water, can go to the plant roots, can go to fruit and veg, or goes to the grass and grazing, and then can go to chronic wasting disease in deer, or obviously BSE in cattle, or whatever else is eating. So potential consumer dangers. So consumption of prion-infected meat, because it doesn't just go into the nervous tissues, what we thought 20 odd years ago when BSE first started, it will actually go into the meat as well. So be wary if you're shooting wild deer in the States, or obviously if it's going to farms. If these jabs were to be used in the farms, we've got the potential that it could go into our human food chain. Via the crops as well, and potentially into the water. And potentially dairy produce as well. The other thing is, which is important because I'll talk about, they're looking at changing the rendering process in proteins, which I'll talk about shortly. It's important to note that the consumption of prions that pass through another species can have accelerated misfolding activity. So for example, if a BSE cattle prion went into the soil, went into the plants, the deer ate it, and then someone ate the deer meat, obviously that because it's a bovine cattle prion, it won't cause disease in the deer. But if someone were to eat the deer meat, which had the BSE prion in it, the activity or the path of what happens in the person who ate it would be accelerated compared to if they'd had the beef itself. So this is an important point to note. So there's a risk of new and previously unknown prions, e.g. super prions, so they wouldn't have any species specificity. It would be across the species barrier with no problems at all. Just as an example of how dangerous they are, previously unconsidered risks from surgical instruments. So they had, with Creutzfeldt-Jakob disease, which is the human version, they actually had some patients where they did some brain surgery on them, and obviously all the surgical instruments get sterilized between operations, and the person who had the next operation following the previous one, they actually got Creutzfeldt-Jakob disease from the sterilized surgical instruments. So that's an example of how indestructible they are potentially. So we've got to have considerations for blood transfusions, organ donation, and exosome shedding as well. If it becomes rife, see, we can take a bit of a breather now. We're not in this situation yet. I'm warning where it might go to, so don't get overly concerned yet. But this is why we need to keep an idea and keep a close view on what is actually potentially going to happen. And also you can get interference with P53, which is the body's own natural cancer suppression. So it's our protective cancer levels that we have, all of us have got it in it. So if you interfere with that, then obviously you can get things called turbo cancers developing. So what's the difference now compared with what happened with BSE back in the 1990s? So if they start introducing these modRNA vaccines for a whole herd inoculation, massively going to increase the chances of producing prions. And it's not really going to be a question of if, it's probably going to be a question of when, simply because the numbers involved make it far more likely. Even if it was a 0.0001%, by the time you've injected 100 million animals, that is actually quite a significant number. It becomes a game of numbers. As I said, they're highly indestructible, but they will tend to then accumulate and concentrate in the environment and also in the animals potentially as well. They're impossible to filter or neutralize them. So as I say, the sheer numbers of animals involved will make it greater the risk. And also bear in mind, with the WHO One Health policy, they can potentially mandate action in animals as well as obviously what they could potentially mandate in or try to mandate in people. So think of the pandemic treaty that they're trying to push through. There is the potential within that it could even affect your animals if they say, right, you've got a zoonosis, which is a disease which can be passed from people or from animals to people. If you've got the zoonosis, therefore we're going to mandate these mRNA jabs, and the WHO could potentially do that. If it gets passed and if it's allowed. Hopefully it won't. And given people's reluctance now to take the modRNA jabs themselves, and given the existing government contracts, animal use could be seen as the contractual solution to this. and could be pushed under the excuse of countering antimicrobial resistance as well. So in the same way that they changed the definition of a vaccine to allow the modRNA, they could say, well, this one, because we're using it against a bacteria, we're going to call it an antibiotic. So just be careful. It's not happened yet, but it is a potential risk down the line. So obviously, a lot of us have got animals and pets as well as our farm animals. So there are already modRNA jabs being used in North America primarily already. So they've got a rabies one in Canada, canine influenza in USA, Mexico, Chile and Philippines, which came in June 2024. And there's a cat FELV, feline leukemia virus, in the USA. It started in February, so just happened in 2025. They're not in the UK yet, but these are made by MSD now, and they're now calling them RNA particle vaccines. So they're changing the name to try and separate out from all the bad press, obviously, the mRNA jabs have had up to date. And they're under the brand line name Nobivac NXT vaccines. That's how they're being branded in the States. Dare say if they came or tried to come elsewhere, they would use a similar sort of brand name. Given that shedding has been proven to occur with the COVID jabs, I wonder if anybody has looked into these for the animal side of it as well. I suspect not. There are also self-amplifying modRNA, where the body is directed to produce the mRNA itself in an uncontrolled manner, which will then produce the protein in an uncontrolled manner. So you've got two levels of uncontrolled amplification potentially going on. Again, none of these will have been checked to see whether they've got prionogenic activity or any autoimmune issues as well. Obviously, when our pets, we hope they're going to live long, healthy lives, so the chances of some of these diseases actually manifesting will be much higher in our pets than we'll potentially see in animals which are being grown over a much shorter time period for meat. They won't necessarily have time to show and manifest these problems. But we would see them in our dogs and cats, unfortunately. What could possibly go wrong? So the consequences. So there's no safe level. What's happened to number 2? Prions and amyloid, which is, again, another protein misfolding. You get cardiac amyloidosis and renal amyloidosis. So again, it's a protein misfolding that happens in other parts of the body. The enzymes can potentially break them down, but the breakdown products themselves can have potentially prionogenic activity too. So breaking them down isn't necessarily the best idea, because all you do is end up seeding it around the body. So potential treatments need to be properly assessed. Don't dive into something just because someone says, oh, this breaks down, prions on the internet. It's like it may do, but just be careful what you do, what the consequences of that are doing. It just needs to be properly assessed. So their resistance to heat, we've already done that one, and all treatments will work for everybody. And the delay in disease onset means that some of these treatments may start too late because it's already reached a certain level of folding before the symptoms manifest. And if you think this is all fanciful and that there's nothing to back it up behind it, I'm sorry, but I've got some bad news for you. They've done a prediction. They've got some actuaries or whatever they do within the pharmaceutical industry to look to see what the future of prion treatment is going to be in the future. And as you can see, from 2023 going up to 2030, it's massively increasing. So they've somewhere have got an idea that prions are going to become a problem in the future, because huge investments will be put into developing drugs to treat these on the background of this. So this isn't my speculation. This is something which the industry themselves are already looking at. Otherwise, they wouldn't come up with this graph. The same with Sandy, everything I put up, I try and make sure that I can back it up with something that is either from their own literature to prove that what we're saying isn't speculation. It's actually got some basis and fact behind it. So this might be a bit of a shock for some people, but the processed animal protein inclusion rules are looking at being changed. So DEFRA has a consultation which is ongoing now, looking on relaxing the PAP rules, and Funny enough, they've chosen April Fool's Day for the consultation to end, so I'm not quite sure who's being played for the fools here. So this would basically allow poultry pap into pig feed and pig pap into poultry feed on the basis that both species rarely show transmissible spongiform encephalopathies. As I've already said, when you've got a prion passing through a species that it doesn't supposed to show in, but you're also chosen a species where it tends not to show, you've got even less warning that something is coming through that process. So what they think is actually a safety net actually may be a hidden opposite. They also intend to allow insect pap into pigs and poultry feed, and ruminant collagen and gelatin in non-ruminant animals as well. And the reason for justifying this is because it brings us into line with the EU. But I thought we'd left that for a reason. That is not a good enough reason to change our regulations. It's got, let's base this on science. If we found that what we did with the protein rendering rules actually worked, then if it ain't broke, don't fix it. It's just common sense. So it seems to me to be the perfect way of potentially developing a super prion, that where we wouldn't necessarily see it until it's already been transmitted to people. Again, what could possibly go wrong?